The ocean is home to many unique and fascinating creatures, and one of the most scientifically valuable among them is the jellyfish, specifically Aequorea victoria. This transparent, glowing species may look simple, but it has contributed greatly to modern science through the discovery of green fluorescent protein, or GFP. In this presentation, we will explore the genetic makeup of Aequorea victoria, including its chromosome number, key genes, and its important role in biotechnology and pharmaceutical research. Its natural ability to produce fluorescence has helped scientists study gene expression, track proteins, and conduct disease-related research more effectively. In this part of the presentation, I will explain about the number of chromosomes found in our selected organism, the jellyfish Aequorea victoria. Aequorea victoria, also known as the crystal jelly, has a total of 24 chromosomes, which means it has 12 pairs of chromosomes in each somatic cell. This chromosome count is referred to as the diploid number, written as 2n equals 24. Although jellyfish may seem like simple creatures, their genetic structure is actually quite complex. Their chromosomes contain all the genetic instructions needed for survival, including the famous gene that produces green fluorescent protein, or GFP. The genome of Aequorea victoria is estimated to contain around 700 million base pairs, making it relatively large compared to other marine invertebrates. The chromosome carry not only the genes responsible for basic bodily functions, but also for the jellyfish's unique ability to glow. Understanding the number of chromosomes is important because it helps researchers study how genes like GFP are inherited and expressed. These chromosomes play a key role in the jellyfish's biology and are the foundation for its glowing ability that has become so important in science today.
Number of jellyfish genes. Jellyfish are ancient soft-bodied marine animals that have lived in the ocean for over 500 million years, making them some of the oldest creatures on Earth. Despite having no brain, heart, or bone, they survive and thrive using a simple nerve net and their bell-shaped body to move through the water. Made up of 95 to 98 water, jellyfish use their tentacle lined with stinging cells called cnidoctids to capture prey like small fish and plankton. Some species like Aequorea victoria glow in the dark through bioluminescence and have contributed to important scientific discoveries such as green fluorescent protein GFP. Genomic studies show that jellyfish typically have around 20,000 to 25,000 protein coding genes, along with hundreds of non-coding RNA genes and a number of pseudogenes, non-functional remnants of active genes. Altogether, the total number of annotated genes in jellyfish is estimated to be between 25,000 and 30,000, depending on the species. They live in every ocean, from surface water to the deep sea, and have a fascinating life cycle that includes both polyp and medulla stage. While most are harmless, a few species can deliver powerful and even deadly stings. These creatures are not actually fish but are classified as invertebrates, and they continue to intrigue scientists and ocean-level labs.
Number 1. Biosensor for drug and therapies. GFP is utilized in the development of biosensor to detect biological activity within cell. For example, GFP is used in detecting protein-protein interaction and measuring enzymatic activity in cell, which is critical for the development of new drugs. In the study of microalgae, GFP is used to monitor the effectiveness of oral recombinant protein delivered to fish, potentially replacing injection therapies. Number 2, detection of ion and pH. GFP is also employed to monitor change in the pH and ion concentration within cell. This is valuable in the studying biological processes in the living cell, which plays an important role in the development of pharmaceutical therapies. In the study, it helped assess how microalgae affect cellular condition in fish during recombinant protein delivery. Number 3, detection of genetic changes. GFP is used in study to identify genetic changes within cells that can be used to monitor the effects of pharmaceutical treatment on gene expression in the cell or organ. In the case of recombinant protein delivery to fish using microalgae, GFP helped track genetic change in fish after receiving the protein therapy, providing valuable information on how treatment impact the organism's genetic makeup. Number 4, use of GFP in drug distribution. GFP is used to track the distribution and effects of drug within the body, providing clearer insight into how drugs interact with cell and tissue, and how they can function more effectively. In the study of microalgae, GFP helped track the distribution of recombinant protein within the fish body, offering deeper insight into the effectiveness of oral therapies compared to traditional injection-based therapies.
So for the next slide, we will look at the method evaluating microalgae as a recombinant protein delivery platform in fish using GFP. So for the sample preparation, we will use the freeze-dried Chlamydomonas reinhardti strain, and for the experimental setup is fish larva fed with freeze-dried algae and GFP fluorescent observed with confocal microscopy at 40 times magnification. For the Western blotting is protein extract from the algae, and we will analyze with Western blot to confirm the GFP expression at various concentration. For the histology and the fluorescent imaging, we will use as a fish feed, and the intestinal tissue cross-section stained to detect GFP, and this image captured at 40 times magnification.
Case study: an evaluation of microalgae as a recombinant protein oral delivery platform for fish using green fluorescent protein GFP. GFP, green fluorescent protein, derived from Aequorea victoria, a species of jellyfish, is used in various applications within the pharmaceutical field. For instance, GFP serves as a visual marker in numerous biological experiments to monitor gene expression and protein interaction within living cells. Its unique factor of emitting green light when exposed to ultraviolet light makes it extremely useful in pharmaceutical and molecular biological research. Case study example. For example, the article discusses a study in which GFP is used to monitor gene expression in the fish cell treated with gene therapy using microalgae as a recombinant protein delivery platform. This demonstrated how GFT technology aids in understanding how specific therapy influence protein production in cell, which can enhance the efficacy of medical treatment. Conclusion: the use of GFP in pharmaceutical provided many benefits in understanding the biological mechanism of diseases, drug effects, and development of genetic therapies. With its ability to detect biological change in real time, GFP is an essential tool in drug research and development, making it invaluable in the pharmaceutical, biotechnology, and molecular biology field. In this study, GFP played a critical role assessing the effectiveness of recombinant protein delivery system via microalgae for fish, opening up opportunity for development of efficient and cost-effective oral therapy.